Hi, I'm Dr. Sherman Canapp with Canine Longevity, and I'm extremely honored to have Dr. Nicole Ehrhart. She is the Director of Longevity at Colorado State University, and this program is translational. They look at aging in humans as well as animals. Dr. Ehrhart, it is an honor to have you here today.
Thank you for having me.
So I know you as the world-renowned surgeon, oncological surgeon, oncology, all things cancer, worldwide. And I'm starting to notice this change as I'm looking into your research and your publications and your work in this whole area of regenerative medicine and then translational medicine as far as aging is concerned and your director position at Colorado State. Can you tell me how did that path begin and where are you now?
All right, great question, a common question. So a lot of the work I was doing in regenerative medicine was looking at acute defects, so these large musculoskeletal defects and how we can regenerate tissue in areas where we've exceeded our regeneration capacity, such as large muscle and bone defects. And then it kind of occurred to me, I did a sabbatical in 2018 at the Steadman Philippon Research Institute and had the opportunity to work with master athletes who are these incredible, people over the age of 60 who are running like 100-mile marathons and other, incredible feats. And I was collecting stem cells from their muscle as they were having various different types of surgeries and recognized that their stem cells were extremely robust as compared to other people that were their age, that were sedentary. And I really started to get the light bulb that perhaps some of the treatments that we've been using to regenerate tissue in acute defects could be repurposed to think about this as a chronic situation and regenerate or rejuvenate tissues that have been deteriorating or degenerating over time. And that sort of leapt me into the whole longevity field, and it was like a perfect match. So it's been an amazing, amazing journey.
And I see a lot of your work says this term One Health. Is that a translational term, or help me understand the One Health term that you use?
Yeah, I mean, we share the planet with these four-legged creatures and other creatures, and they live in our environments, and they are part of our everyday life. And, it's important that we think about this as a paired situation. A lot of times the animals that share our environments are sentinels for our own aging pathways. They're exposed to the same things that we are. And not only that, they have a shorter lifespan than we do. So many of the interventions that we might use to improve health span and longevity in humans can be studied in companion animals and other animal species in order to, in a shorter period of time, in order to see if they're efficacious or not. And so why not use that as a two-way street, right? What can help us can also help our pets and vice versa.
So you speak of efficacy. Have you seen, or can you share with us some of the significant findings you've had clinically or even from a benchtop research standpoint?
Sure. I mean, we are in early stage research with a lot of these things, and the idea is to translate this into intervention trials in companion animals that then will inform human medicine. And the idea is really to accelerate and de-risk these longevity and health span interventions. And so a lot of the work that we're doing is in the stem cell world, which shouldn't surprise you if you know I've been involved with regenerative medicine all these years. The main thrust of our research is in something called extracellular vesicles or exosomes. And these are the things that the stem cells secrete in order to create their biologic effects.
So do you feel, or have you seen then, that we can take the exosomes, so the stem cells are releasing them, is there a way that we concentrate them, or how does that work as far as having a clinical application?
Right. So the parent cell line, in the case of the research I do, are the mesenchymal stem cells. And they're cultured outside the body, and then we collect the supernatant or the culture media that in which these exosomes are secreted by the MSCs. Then we concentrate those down using a process called ultracentrifugation. Then we can count them and know how many are in there. And then our research has been looking at what is the cargo content and can we optimize the cargo for specific applications. Things like muscle loss or tendon healing or bone healing. And the conditions of the parent cells and the type of parent cells that you use actually influence the cargo that is within the exosome. So it's tunable. You can actually tune it for specific applications.
So then would the idea be that you can concentrate this down? Would you be able to keep it, say, in cryo or keep it preserved somehow, and then as needed, you would be able to dose that out? And is it to the same patient, or would you be able to use that to other patients? Of course, that has to
Great
go down
question.
the FDA line, but is that something you're looking into?
Yeah. So one of the advantages of doing the exosomes or extracellular vesicles over a cellular therapy is that they can be frozen and they remain relatively stable when frozen for long periods of time. There's even evidence that they can be freeze-dried, which would be great because then you don't need cold chain storage and you could ship anywhere. And also because these can be donor cells and they're non-immunogenic when they're formed, so these exosomes themselves don't create an immune response, a donor can be used to treat multiple different patients rather than the individual patient donating back to themselves. And so yes, there are all the regulatory pathways that we have to navigate, but the promise here is that we can have an off-the-shelf product that could be widely clinically applicable to a number of different disease states and a number of different patients.
So I have to ask you, because you're dealing with aging and longevity, there's this theory or hypothesis, I would say, that dogs age at this 1 to 7. You know, we always say, oh, 1 year in people life is 7 years in dog life, or maybe it's different in small breeds versus giant breeds. Is there any truth to that, or is there anything you've looked at? I know it's translational, so you'll get a human in animals, but what can you tell me about that?
Well, I haven't looked at that personally, but there's been some very eloquent research done in that area. And specifically, they're looking at methylation clocks, or the little tags on our DNA that change as we age. And the person that's done probably the most work in comparing animal, specifically dog age to human, is a guy named Trey Eidecker. And he published a study that got a ton of press, and I think it was, 4 or 5 years ago, looking at the age comparison between humans and dogs based upon this methylome or these DNA tags that they can measure. And it turned out that it's not exactly 1 to 7. There's an exponential curve that changes, but we do know that dogs go through all the same developmental phases as people, and we know what ages those are. You know, infancy, adolescence, adulthood, senior, life. And so we have scales that we can actually apply that are really applicable from the standpoint of the methylation and the biologic aging of the species that we're interested in this case, dogs.
Oh. Biomarkers. So some of the work I think you've been doing is on biomarkers. I know there's so much out there right now as far, inflammation, gut inflammation, and GI mapping, and even its effect not only on aging but cognitive function with inflammation and markers. What are you doing in the world of biomarkers? Is that something you're looking at within your studies?
We are. We're really interested in this whole idea of the peripheral central communication and brain health and what affects brain health. For example, we know that certain lifelong interventions like exercise and good sleep are related to longevity and healthy aging in people, right? And for a long time, we weren't really sure how that happened. We also know that exercise is preventative or protective against cognitive decline in people. And the Dog Aging Project has shown the same thing in dogs. So there's something about the periphery that happens during exercise, as an example of one intervention, that impacts our central nervous system. And so what we've been looking at are the exosomes that circulate in our blood that happen after we exercise, and how those are actually dampening inflammation within the brain, both in induced inflammatory models as well as like natural aging models like the dog, and we have certain other animal models like guinea pig and other, where we know those naturally age and they develop cognitive decline over time, and we can intercede with different exosome interventions.
Wow. There's also something I've read about in your lab. It's the Human Dog Biobank, I believe it is. Can you explain that?
Yeah, so we're really interested in the idea that the animals that share our environments are actually sentinels of our own aging pathway because of their exposures and because they're also, impacted by our lifestyle choices, like our, exercise and active lifestyle versus secondary or sedentary lifestyle or secondhand smoke or other things like that. And so the idea there is to actually have human-dog pairs living in the same household that are providing biospecimen samples over years at a time and pair that with metadata from their medical records to understand what exposures and environmental impacts and even social impacts have on our own aging pathway. And we believe that by looking at changes that might happen in the dog, we can actually preempt or detect changes that might be happening in the human that's living with that dog. Yeah. And so that's a unique part of the biobank idea, is to have the dyads or the pairs together. And I don't think that exists anywhere else in the world.
No. You spoke before about the exosomes and how we can concentrate those down and keep them in cryo. Let's talk about stem cells. So the stem cells age as we age, I believe. Is that correct? And so is there any thoughts then, if that's the case, that you would collect tissues from dogs when they're young and process, place in cryo, so that years down the road when they're older, 10 years of age, they don't have the 10-year-old stem cells, they have their young dog stem cells. And does that mean that they'd be a little bit more potent as far as tissue healing or regeneration? Explain that. Is that something we should be considering clinically, or?
I mean, it's a good idea. It's kind of the idea behind cord blood that's collected when a baby's born, is that you'll have that forever, and that person will never be that age as an infant again, right? And we do know that our stem cell pool diminishes over time as we age, and the robustness of the stem cells that exist also diminish. So in theory, it makes sense to bank stem cells earlier. I think we're starting to get over that hump where we may not need to bank them as much as we may be able to genetically reprogram stem cells even late in life to behave more like young stem cells. And then the other thing we've been working on in our lab is actually exercising those stem cells outside the body. So creating an exercise mimetic, we call it, in a bioreactor, and then collecting the exosomes from those that actually have the same content as a younger animal or younger person, and developing therapeutics out of that.
So we talk about the pillars of longevity and aging: exercise, nutrition, cognitive function. And cognitive wellness is a huge part of that. And I looked further into your research, into your lab, and you have a psychologist, I believe, on your board as well. What type of work are you doing looking at cognitive function and specifically in dogs?
Right. So we're looking at different interventions, both lifestyle interventions and therapeutic interventions that may impact cognitive health as we get older, and also looking at different nuanced ways to measure cognitive health and biomarkers of cognitive health. So our psychologist is a person who's an expert in neurodegenerative diseases. She's a neuroscientist, and she does a lot of work in dementia. She's also someone who runs a center called the Aging Center of the Rockies, which is a center that does neuropsych evaluations on people. So again, nuanced ways of actually diagnosing cognitive decline when it's early and then intervening. And then we're trying to utilize the same types of measurements that are proxies for similar types of behaviors in dogs. And there's many assessment scales that are used for cognitive decline in dogs, and those are very validated and good assessments. We're trying to add to the body of that literature and then pair that with biomarker changes where we can see over time changes in cognitive health and actually intercede early enough that we can maybe prevent rapid decline or further decline.
Oh. So in your lab, what's next? What's your next steps, or what's the next studies or research that you're looking into? What's kind of the hot thing that you're going to be diving into?
Yeah, I mean, I think there's so much parallel work going on looking at different interventions, that one of the things I think we need to advocate for broadly is the idea of creating a very well designed infrastructure for clinical trials for longevity interventions in companion animals, and utilize that again to inform human health, to accelerate and de-risk some of the newer studies. So thinking about ways in which we can add to the body of science, which is huge in the rodent and other preclinical models, but really has failed to translate very rapidly into people. And I think the dog is the bridge. I really think that if we can create a very well-designed infrastructure that's multi-institutional, that we can perform these kind of compliance studies, GCP compliance studies that are multi-institutional, that are placebo-controlled and double-blind, we can probably move the needle much faster. So that's the kind of thing I really want to develop from the standpoint of clinical translation. And in the lab, we're continuing to develop other things like other exosome-based therapeutics, as well as different interventions that improve longevity and cognitive health.
You're looking at biomarkers and looking at something where we have metrics that we can look at translationally and look at other universities, and having those would allow us then to look at other medications or drugs that are out there. We talk about metformin, we talk about rapamycin. It would be nice if we had those objective outcome measures that then we could look at all those different types of products that they're looking at for humans and start applying those to dogs. Of which, have you done any work with rapamycin in dogs, or do you know anything about, should that be something that, I believe there's some studies, the Dog Aging Project, I believe is one of them. Can you say anything about rapamycin for dogs?
Right, and I haven't done any studies personally with rapamycin in dogs, but I think the Triad study in the Dog Aging Project is the most active one currently, and they are currently enrolling I think that there are a number of different other studies that could be done in different populations. I'm very interested in the cancer population. Yeah. I'm particularly interested in how the treatment with chemotherapy actually accelerates aging. We know this in people. We can see it. We can observe it in people. Yeah. And we believe that also happens in dogs. And so we can look at some of these longevity interventions that might target, for example, senescent cell burden or other things that may actually impact cancer outcomes. So if we can rejuvenate the immune system and prevent a lot of senescence buildup in the immune system, perhaps we can actually have better outcomes with our cancer treatments in our pets as well, and then of course in humans. So that's another area of research I'm super interested in. I haven't done any specific work on rapamycin, but that would be a therapeutic agent that would make a ton of sense.
Yeah, completely.
Yeah.
So if you were going to say to any of the pet parents that are out there, anything they could do to help currently or in the future with the wellness, longevity, health span of their particular pet, give some advice, give some recommendations on things that you just think would be easy that they could consider straight away for their pet.
Well, we know across all species that have been studied that keeping a lean body mass is important for healthy longevity. We also know that in dogs, at least based on the Dog Aging Project observations, that having another dog in the household is generally a really health-promoting thing into old age. And then, of course, exercise, active. So those are just interventions that I think anyone can do. It's not easy to calorically restrict your dog all its life, especially if you have a Labrador, which I have too, so trust me, I know. But the type of things that we can actually mimic caloric restriction with certain therapeutics, those are things that I think are going to come online in the near future. So as a pet parent, what I would do is stay informed about longevity trials that might be available to you in your location. Keep in tune with the media and scientific literature. Ask your veterinarians about this, because I think so much is coming online that will really impact the health and longevity of our pets in the near future. And so we're starting to see this explode. It's a very exciting area of research, and I love what you're doing. I'm excited that you're asking these questions, and I hope that pet parents can continue to stay informed both through this program and, keeping in touch with scientists like myself who are working at the front lines.
Dr. Ehrhart, can you share any clinical cases or anything that comes to mind on a case, we'll call it a successful case, that really you remember?
Sure.
As it relates to, say, regenerative medicine?
Right. Yeah, I mean, a lot of the work that we did early on was in limb salvage, right? And this is where you have to remove a large portion of bone usually, or sometimes bone and muscle, and you're trying to get that area to heal. Some of the things that we've used to heal the area are things like bone grafts and even implants. And one of the areas that was a really exciting area where I felt like we really made some progress was in looking at the integration of these implants with the parent bone. And so we started thinking more along the lines of the biology of the patient. And, instead of creating better and better mechanical parts that fit better or function better, how do we harness the biology of the patient? And so we started with stem cells. And in a particular patient, this dog had an osteosarcoma. We also wanted to make sure that it was safe to give stem cells to a dog with osteosarcoma. And it turns out that we did a bunch of mouse work and looked at that very carefully, and we felt that it was safe. In this particular patient, the owner was really willing to go the extra mile to hope that this dog would heal, and we used stem cells to help his limb salvage implant actually integrate with his parent bone. And that dog was one of the few successes I've had. Now that's an N equals 1. Yeah. I'm not going to try to tell you that this is going to be a treatment that will work for everyone. And certainly we've looked at different ways that we can optimize that and tweak that. But these are the kinds of people that are looking for a solution, that are willing to go the extra mile, that are willing to be part of something that's on the cutting edge. And those are exciting times for us because we get a chance to really see if what we're seeing in the laboratory is really going to work in real life. And there's been a lot of progress in that area. We talked about the limitations of stem cells. I think the extracellular vesicles or exosomes will behave the same way, and yet they're so much easier to work with. So that's probably our next step. And again, getting that into our patients is the most important part of this. The science is great. We can learn a lot about that, but until it becomes a real-world treatment or a real-world benefit to those animals, it doesn't really ever have the impact that we're really looking for. And as a clinician scientist, that's what our work is really all about.
I think that's a great example of allowing the body to help heal itself.
That's right.
Dr. Ehrhart, I'm so excited about what you're doing and so fascinated on the lab, everything you have so far. I'm just dying to continue to follow you.
Thanks.
This is Dr. Sherman Canapp with Canine Longevity. Remember, it's all about canine health span, not just lifespan.
Good girl, Fachi.